Hey everybody, welcome back to General Biochemistry Lecture. We are jumping to chapter 25 and talking about DNA metabolism. I know we use the word metabolism for a lot of things. In this case, what we're talking about is how do we go from having one DNA molecule to replicating that DNA molecule and having a copy of it. So don't get too, excited and worked up. This is stuff that you've already encountered in biology class. We're just going to take it a little bit deeper. Before we get into the actual process of replicating DNA, we've got to talk about gene naming and protein naming because we're going to see some kind of wildly different systems when we look at bacteria versus eukaryotes. So with bacterial gene naming, typically you're going to see three italicized lowercase letter, and you're going to see that the function of the gene, aka the gene product, is the name. So DNA, UVR, REC, they're usually, acronyms for things. Capital letters that are added to an abbreviation reflect the order of discovery, not the order in which these products operate. So DNA A, B, and Q were discovered in that order, A, then B, and then later on Q, right? But they don't have to operate in that order. They could all operate at the same time doing the same, something similar, or they want DNA Q, that product could operate first. So it's really about when it was discovered, not the order in which it's used in a process. With protein naming, it's typically the gene name and you remove the italics, you capitalize the first letter. So if you have a gene, DNA A, there's the italics here. Instead of having the italics, you remove it and you also capitalize the first letter. So this is just to help you recognize when you're looking at a gene name versus a protein name. This is for bacteria. Eukaryotes, there is no single convention for all the eukaryotic systems. So in yeast, gene names are three italicized uppercase letters followed by an italicized number. Protein naming is pretty much the wild, wild west. So it's going to vary from system to system. Sometimes you'll see that the gene name is reflected in the protein name. Sometimes it'll be something completely different. It's complicated. I'm sorry. In yeast, you have some proteins that have long common names like cytochrome oxidase. But there are other proteins that are named the same as the gene, so just like in bacteria, only they have the letter P after them to emphasize the fact that it's a protein. So for example, there's the RAD51 gene, and notice that it is in all italics, capital letters, and then the protein, you just have the one capital letter, there's no italics, and the letter P follows it to say this is the protein, y'all. You're going to see a variety of things when it comes to eukaryotic protein names and gene names. Unfortunately, we just ain't organized like that. But with bacteria, it's pretty simple, and we're going to focus on looking at the bacterial system first, and then making comparisons to the eukaryotic systems. So here are the key principles for this chapter. We're only covering the first two sections on replication and repair of DNA. We're not talking about homologous recombination. So let's start with DNA replication. Again, you've heard of this, you know the process by and large, but we're just going to add to your knowledge. DNA is a template. Okay, a template is a structure that allows molecules to be lined up in a specific order and joined together to create a macromolecule with a unique sequence and function. The structure of DNA reveals that one strand is complementary to the other, and each strand provides the template for making a new strand. So we replicate both strands of DNA because they both hold valuable information that we need in order to, create a new cell when we split, do mitosis. And you need all of that genetic information to make it into the next generation of cells. There are basic principles that apply to DNA synthesis in every single organism. And we'll talk about these kind of as we go through the process. So DNA replication is semi-conservative. We're going to just define what that means in a second. Replication begins at an origin, and it usually proceeds bidirectionally. So you're not just starting in one place and moving only to the right. You're going to be moving in both directions, and you're going to be replicating all the DNA kind of simultaneously. DNA synthesis proceeds in a 5' to 3' direction, and it's semi-discontinuous. You'll understand what all three of these points mean by the end of this section. Let's start with talking about what semi-conservative means. Each DNA strand serves as a template for the synthesis of a new strand. And when you're finished with replication, you've got 2 new DNA molecules. Each one has an old strand and a new strand. So semi-conservative means that little bit of old, little bit of new. That's what we mean by that. Replication begins at an origin, and we said that it proceeds bidirectionally. Right now we're looking at a bacterial chromosome, which is a plasmid. It's double-stranded circular DNA. Now, our chromosomes are a bit more complex, and we'll talk about that towards the end of this section. But right now, we're just focusing on bacteria. So we have this parental duplex meaning we've got 2 strands. And then, when we're ready to replicate, there's what's called a replication fork. And this is a dynamic point where the parent DNA is being unwound and separated, and the strands are being duplicated. So remember, DNA is wound up, and plasmids are usually supercoiled. So they kind of look like that. That's a supercoil. When you go to replicate in bacteria, you first have to unwind and establish some kind of a replication fork so that all the machinery that you need can actually bind to the origin and get things going. Both of the strands are simultaneously replicated. So we're not going to do one strand and then the other. That would take way too long. Both ends of the bacterial chromosome have active replication forks. So what that means is that we have bidirectional replication. So we know that all of this is happening bidirectionally, and we are replicating both strands at the same time. But how does the cell know where to start replication? We're not actually starting in a random place. There's an origin. So there's something that you can do called denaturation mapping, and that's where you selectively denature or unravel. We talked about denaturation in terms of proteins, but you can do the same thing with DNA and RNA because they have structures associated with them. You can denature sequences that are particularly rich in AT base pairs, and that kind of provides landmarks along the way for your DNA to know where you are. And you can reproduce a pattern of single-stranded bubbles. This is kind of what the origin looks like, right? And the origin is the location where replication loops are initiated. They are particularly rich in AT base pairs, and this is where you can have the separation of the two strands to enable all of the machinery to bind that you need for replication. New DNA strands are always synthesized in the 5' to 3' direction, and the reason for that is because you need the 3' hydroxyl to elongate your strand. So it is involved in creating that phosphodiester bond. Okazaki fragments, which we're going to talk about how they're made, they are short DNA fragments that are synthesized in the replication of one of the new DNA strands. It's going to be the lagging strand. This may sound familiar to you if you remember DNA replication from biology. These Okazaki fragments are made in bacteria and eukaryotes. In eukaryotes, they're kind of short, like 200-ish nucleotides long. In bacteria, they're like 1 to 2,000 nucleotides long. So a little bit of a difference there. And once all of those fragments are made, they are sewn up by DNA ligase. Now we'll define the leading and lagging strands. So the leading strand is the strand that it's synthesized continuously. It proceeds in the 5' to 3', and it's happy. The lagging strand is the one that's a little bit more complicated. So the leading strand replicates in the same direction as the replication fork. It's continuous. The lagging strand is still in the 5' to 3', but instead of being continuously made, they're discontinuously synthesized by making all of these little Okazaki fragments. And the overall direction is 5' to 3', but that actually moves in the opposite direction of the replication fork. So there's some more gymnastics and coordination that needs to occur to make sure that the synthesis of both of the new strands actually happens in tandem. So new DNA strands are always synthesized in the 5' to 3' direction, and the template is read in the 3' to 5' direction. So the leading strand, so in blue we've got our old DNA. The leading strand is the one where you have 3' to 5', and you're just synthesizing the complementary 5' to 3'. So there's no issues here. Our replication fork is moving from left to right. So this big red arrow shows you the direction of the movement of the replication fork. So eventually, you're going to get all the way down to here. However, the lagging strand, it's 5' to 3' because it's complementary to the leading strand. So how we get around that is by making all of these tiny fragments that start here, and then we work back towards that 5' end. So the replication fork is moving from left to right. However, on the lagging strand, the replication is actually from right to left in these short fragments. So eventually, you cover the entire strand, and then you have to sew up all of those little spots in between using DNA ligase. Before we can talk about how DNA is replicated, we have to talk about how DNA can be degraded or destroyed. And the enzymes that do this are called nucleases. They degrade nucleic acids. DNases specifically degrade DNA, but there are nucleases that are not specific for DNA or RNA. There are also RNases, and we'll talk about those in chapter 26. Exonucleases degrade nucleotides or nucleic acids from one end of the molecule. So they have one direction that they operate in. They either operate in 5' to 3' or 3' to 5'. Endonucleases, what they care about is a specific sequence within that nucleic acid. So they will chew up specific internal sites. They'll make cuts. And these enzymes are used in the lab. We also call them restriction enzymes or restriction site enzymes. And we use those to make specific cuts when we're trying to kind of cut and paste and make DNA. DNA is synthesized by DNA polymerases. So oftentimes you will see abbreviations for this long-term DNA polymerases. So I abbreviate polymerase with pol, and I'll also say DNA pol, RNA pol versus, polymerase. And there are lots of polymerases. So if I'm referring to a specific one, I'll say DNA pol 1 or pol 2 or pol 4, etc. So DNA polymerases are complex enzymes that can synthesize DNA. They usually have additional activities aside from sewing together nucleotides. E. coli have at least 5 DNA polymerases. And we're going to start talking about these polymerases and their functions. Before we get to looking at the different DNA poles, we're going to talk about the reaction a little bit. So the reaction is a phosphoryl group transfer. What we're looking at on the right is the active site of a DNA polymerase. Notice that there are 2 magnesium ions. There's a purpose. One of the magnesium ions is required to help make the 3' hydroxyl a better nucleophile. That the oxygen in the 3' hydroxyl is going to attack the alpha phosphate, which is the one that's closest to the pentose sugar. It's going to attack that phosphorus atom, and we're eventually going to get rid of the beta and gamma phosphates, so the last 2. The second magnesium ion stabilizes these last 2, this diphosphate, as a leaving product or a leaving group. So it makes it okay for it to leave. Okay, it's all right. It's all right, diphosphate. You can go now. So you should be able to draw this reaction. It's just a plain old phosphoryl transfer. So just keep that in mind. This is something that you've drawn before. It's just with different substrates. So again, we've got the two magnesium ions. One helps to deprotonate that 3' hydroxyl, make it a better nucleophile. And the other one helps to bind the incoming deoxynucleotide, and it helps facilitate the departure of that pyrophosphate. So in general, what happens here is we have a deoxynucleotide monophosphate, and we add to it. So this is the DNA, right? And then this is our new nucleotide. What we end up with is a lengthened DNA chain, so N+1 here, and we release that pyrophosphate. Now, typically speaking, that pyrophosphate is then cleaved by pyrophosphatase. We've talked about that before, and that reaction releases energy, which helps make reactions more favorable. It helps provide the energy to do reactions. And what you'll see as we talk about the process of replication is that replication is expensive, and so is repair. But because DNA is so crucial to the survival of the organism, you have to make sure that your replication process is robust, meaning that you can do it well with high fidelity, and you have to be able to maintain that information source, which is where DNA repair comes in. Okay. So the reaction itself actually proceeds with a minimal change in free energy. You've got a phosphodiester bond that's formed, and it replaces the phosphate anhydride that is what links all of the phosphoryl groups in the deoxynucleotide. The base stacking and base pairing interaction stabilizes the DNA product, and the formation of products is facilitated by, again, like I said, the hydrolysis of the pyrophosphate. So that release in energy helps drive the reaction. Polymerases require a template. They can't just bind to naked DNA and go. They need some kind of a template, and that template strand has to bind, it has to follow the base pairing rules, right? So there's a primer strand that's added, and then the polymerase, the DNA polymerase, can come in and recognize, oh, okay, yeah, I got this from here. And it will go and continue on adding additional nucleotides that base pair properly. There are some different spots that we need to recognize on the pol enzymes. There's the insertion site, which is where you're going to be adding a new nucleotide. And there's the post-insertion site, which has the base pair that you just finished making. So here with the primer, we've got the primer terminus that serves as the, it holds that post-insertion site when the pol is first binding to the primer strand and the template strand. So it recognizes the end of the primer. Oh, okay, this is a GC base pair, great. And now I see that there's a T, so I'm going to allow an A to bind, and then I'm going to seal the deal. Then once it does that, it's going to slide or translocate one base pair down so that what you just formed, that AT base pair, moves from the insertion site to now it's at the post-insertion site. And you make way for another incoming DNTP, and that's the new insertion site. So just in case you didn't know, a primer is a strand segment with a free 3' hydroxyl where a nucleotide can be added. It's got to be complementary to the template, and many of these primers are actually RNA. So we start off with an RNA primer, then a DNA pol comes in, and we'll start adding to that. Then at the end, we have to come back around and get rid of that RNA primer and replace it with DNA. The primer terminus is just where the free 3' end is of the primer. The insertion site we already talked about a little bit, but now you'll be able to kind of see it in 3D form. So DNA polymerases kind of look like a hand that's kind of cupped over. So if you take your hand with your palm facing up and you kind of bring your fingers towards you to make kind of a cup in your hand, like a C, and then you turn your palm so that it's facing down towards the ground, that's kind of what a DNA polymerase looks like. So there's kind of this cup region where you have all the binding sites, right? So there's the palm, the thumb, and the fingers. And that's usually how, if you read literature about replication or DNA polymerases, they talk about the structures and refer to these different sections as the palm, the thumb, and the fingers. The insertion site, that's where you have, that's the active site. So the reaction that we talked about where the 3' hydroxyl attacks the alpha phosphorus, and that's part of the first phosphate group on the incoming DNTP, that's where all of that action is happening, at the insertion site. The post-insertion site is the part of the action site where the new base pair is that you just formed when the polymerase slides forward. So you can see that it's almost literally like the DNA polymerase is holding on to the DNA. Now we're talking about this polymerase sliding down the DNA molecule. What this is called is the enzyme has what's called processivity. So if you are a processive enzyme, that means that you hold on to your enzyme or to your substrate and you move along that substrate and you catalyze multiple reactions in a row without releasing the substrate. When we talk about replicating things like DNA or making RNA, or even when we talk about making proteins, you're going to see that all of these mega complexes are processive. They have processive enzymes that hang on to the substrate and slide on down, and they will, just keep on working. Processivity is the average number of nucleotides that are added before a polymerase dissociates. This amount varies greatly among DNA pols. It can go from a few nucleotides to thousands of nucleotides. And what we'll see is that these enzymes on their own have more limited processivity, but when they are in a complex, it enhances their processivity so that it makes the process of DNA replication not take forever. Now let's talk about how accurate replication is. It has to be accurate because this is the information that we're using to, keep us alive, right? And E. coli care about that too. They may not be multi-celled organisms, but bacteria do kind of care that their DNA is, accurate. So errors occur in every one in, we're talking about like billions of base pairs. So every 1 to 10,000 replications, you have an error. The active site of DNA polymerases excludes base pairs with incorrect geometry. So it's hard to get a match of an adenine with something other than a thymidine. But it can happen. But this doesn't account for all of the accuracy of replication. So they will actually insert an incorrect nucleotide every 10 to the 4th to 10 to the 5th correct nucleotides. So you can have tautomerization of the bases, because remember, we're talking about chemicals in solution. And whenever there is resonance or there's some kind of, you can lose a hydrogen, gain a hydrogen, all of those things are happening. So the polymerase has to not really identify, but it has to allow the binding of all of these different molecules, because remember, it's got to allow A, T, G, and C to bind. So it has to have an active site that can accommodate all of those things. And it has to make sure that the base pairing is correct based on the geometry of the base pair. But sometimes you can have tautomerization that causes one nucleotide to kind of spoof looking like another, and it allows that in and will form a bond. So we've got an example of correct base pairing, G to C, A to T. But there can also be incorrect base pairs that still kind of fill the pocket and allow for, the DNA polymerase to be fooled. Now, these that are shown don't really do that, but there are some incorrect base pairings that the DNA polymerase can't really tell. But if you compare how the molecules interact with the enzyme, so the enzyme is kind of like in that beige color, beige tan, and then you've got the nucleotides here. And you can see how there's hydrogen bonding in specific sites that are different from G to C base pairs versus A to T base pairs. So if you have, a group that's sticking out, then it's probably not going to be allowed to stay in the active site. So this G to T base pairing is going to be, thrown out. No, we're not going to bond with you. So translocation of the enzyme is actually inhibited when there's an incorrect nucleotide added. It's not going to slide forward until the right thing is added. And many of these DNA poles have intrinsic 3' to 5' exonuclease proofreading activity, which means that they add something and then they're like, is this the right thing? Yeah, okay, it is. And then it'll keep going. Otherwise, if it says, no, this isn't right, then it can actually get rid of the improper nucleotide match and put in the new thing. So on the right, that's what we're showing here. So let's say that we added a C opposite of a T, and we know that's wrong. The endonuclease site, which is a different site from where you have the reaction going on where you're adding on to your DNA chain, the exonuclease site recognizes this mismatch. And the polymerase is going to reposition itself so that the base pair that's wrong is going to be in that exonuclease site. You get rid of that base that's wrong, And then you move that 3' terminus back to the polymerase site so that you can add the correct nucleotide. So when we say proofreading, we're talking about DNA polymerase activity that will also replace incorrect nucleotides. And that's expensive because we're talking about getting rid of 3 high energy bonds. So we are getting rid of a lot of things to make this work because we've got all of the base pairing going on, we've got the stacking interactions, and we've also got to break that phosphodiester bond to get that nucleotide out. That takes energy. So after you do all of the base selection and proofreading from the DNA polymerase, we're still making an error every 10 to the 6th to 10 to the 8th bases. So there must be some kind of mechanism that enhances the accuracy of replication. And where that comes from is DNA repair mechanisms. So now we're going to focus really on E. coli because that is where a lot of the information of bacterial replication comes from. And E. coli, they're a great little system to use in the lab, easy to use, very easy to manipulate DNA and make changes. So E. coli have at least 5 DNA poles. DNA pole 1 is very abundant, but it's actually insufficient for replication. So the rate at which it adds nucleotides is 600 nucleotides per minute. But that's too slow based on the movement that was observed for the replication fork. It has low processivity too, which means that it only will catalyze a few nucleotides and then it'll fall off. And then another one has to bind, and that's kind of a lot. And all of that put together suggests it's probably not DNA pole 1 that's doing the replicating. The primary function of DNA pole 1 is actually to clean up during replication, recombination, and it helps with DNA repair. So it's one of those enzymes that comes around and says, oh, hey, you got a problem? I've got a solution. But it doesn't actually, it's not the workhorse of replication. So now we'll look at DNA poles 2, 3, 4, and 5. So pole 2 is involved in DNA repair. Pole 3 is the principal one used for replication in E. coli. Poles 4 and 5 are involved in like kind of an unusual form of DNA repair. So we're going to focus on pole 3 for the remainder of this chapter. This is a comparison of the 5 DNA polymerases. This is really just for your entertainment and enjoyment. You don't need to memorize this by any means, but this tells you the gene, the number of subunits, or at least the number of different types of subunits, because you could have, multiple copies of one subunit. It tells you the molecular weight, whether or not there's proofreading activity, and all kinds of things like that. Also the processivity. So you don't need to memorize this, but if I gave you information from this chart, you should be able to parse it and understand what it means and kind of compare one enzyme to another. So pole 1 has 5' to 3' exonuclease activity, which is different from 3' to 5'. We're talking about 2 different directions. 5' to 3' is the front end of the enzyme, and it does Nick translation. We'll talk about what Nicks are in a second. Mild protease treatment, which means that you just kind of gently introduce some cuts into your polypeptide, can separate this domain from the remainder of the enzyme. So you can actually have two functional domains from treating DNA pol 1. Now, nick translation, that means that there's a break or a nick in the DNA, and it's moved along with the enzyme. So let's look at our figure on the right. We've got pol 1, and this is what it does. We've got pol 1 binding to a nick region. This gap here is a nick. We're going along. We've got a new, and we're inserting something here. We're adding on to our chain, and we reach a nick. What happens here is this exonuclease activity that's in this domain here. It's actually going to chew up the nucleic acid in the area that's right after the nick. So the purple is going to be destroyed, essentially. And then, once that happens, DNA pol 1 is going to go in and fix it, essentially. It's going to go in and add on to that chain. This kind of activity is important when it comes to DNA repair, but also in the removal of RNA primers during replication. Now we'll move on to DNA pol 3. DNA pol 3 is huge. Lots of subunits, and as you can see, they all have different functions, and we're going to talk about all of these. So you don't have to memorize, molecular weights and how many subunits per holoenzyme, but you should be familiar with the subunits in terms of what are the different functions that are in this DNA pol 3 holoenzyme. Remember that holoenzyme, we're talking about the whole, fully functional enzyme with all cofactors, coenzymes, all that kind of stuff. So there are some core functions: the polymerase activity. Then there's clamp loading, which we're going to talk about all of these. There's also some regions that are involved in interacting with other factors that are bound to the DNA while it's being replicated. And there are also other factors that are involved in keeping the enzyme itself intact with all these different subunits. So some of these subunits are simply linkers. And then there's also the clamp itself that is required for optimal processivity. And it's literally something that looks like a clamp that allows the enzyme to hold on to the strand and slide across it. So understand the different functions present, but you don't have to know necessarily, the core polymerase is made up of the alpha and the epsilon and the theta. We're not going there. But I do have that information for you because, sometimes you just need it. Some people need that extra detail to make things work. But I'm not going to ask you on a quiz or an exam, what is the purpose of the alpha subunit? No. So the core polymerase has the polymerase activity, proofreading activity, and it also has limited processivity. We've got a couple of other subunits that are the clamp loading complex, which load the beta subunit that allows it to clamp onto the DNA. The entire thing is an assembly of 16 protein subunits. There's 8 different types. So we're talking about a huge, huge enzyme. When we add the beta subunits to this enzyme, so notice how there's a DNA pol 3 with the asterisk. That is different from the holo enzyme, which has the beta subunits. Those are the clamps. So this is a cartoon and a structure of DNA pol 3. When we look at the figures that follow for replication, you're mainly going to see this cartoon here. The beta sliding clamp is very important here. And notice that we've got actually 3 different kind of whole enzymes here. So this, in and of itself, is able to do DNA replication. But we've got 3 different copies of it. And we've got this clamp loader that we're going to need. You'll see why in just a minute. But this thing is huge. And in the center of all of this craziness is the molecule that you're replicating. So this is an image of the clamp. And for each of these, you've got DNA that's looped through it. And that allows for the molecule to, the enzyme to be able to literally slide across the DNA. So, as you can tell already, we need a lot of enzymes and protein factors for all of this to happen. And the entire system is called the DNA replicase system, or the replisome. And it's got more than 20 different enzymes and proteins. So it is massive. Some additional things that we need. We'll talk about the main classes of enzymes. We need helicases. Helicases help to separate the DNA strands, and they can also move along DNA strands. They require ATP. So we'll see where all of the energy expenditure comes in when it comes to making DNA molecules. We've also got topoisomerases that help to relieve the stress that's created by separating these strands. There are also DNA binding proteins that stabilize the separated strands, and you may see these also labeled SSBs, or single-stranded DNA binding proteins. There are also primases, and these are the enzymes that synthesize the short RNA primers that allow for the DNA pol to come in and say, oh, okay, I can do this, and then continue adding on nucleotides. Then, of course, we've got pol 1, which removes and replaces the RNA primers. That's one of its functions. And we've got RNase H1, and that's a special type of nuclease that will degrade the RNA in an RNA-DNA hybrid. It can also remove RNA primers. So we actually have both of these. Then we've also got DNA ligases, and they seal up the nicks in the DNA backbone after you remove and replace that RNA primer. And they also seal up all of the little gaps in between the Okazaki fragments. There's 3 stages that we think about when it comes to DNA synthesis, and you'll actually see this throughout the central dogma. So replication of DNA, making the messenger RNA transcript, and with protein translation. There's initiation, where you're kind of getting set up. There's elongation, where you're just chugging through and adding nucleotide after nucleotide. And then there's termination. Once you reach the end, you need to get all of this machinery off, and you need to also separate your two different DNA. In this case, they're plasmids that are kind of entangled together. So all of that needs to happen, and that's termination. We'll start at the beginning. It's always a good place to start. We said that replication always begins at an origin. And in E. coli, the origin consists of 5 repeats of a 9 base pair sequence. These are called R sites, replication sites, that serve as binding sites for the key initiator protein DNA A. So remember, we're talking about bacteria. So capitalized first letter, lowercase for the following 2, followed by capital letter. That's going to tell us it is a protein. There's a DNA unwinding element, abbreviated DO, and that's a region that is very rich in AT base pairs. So remember that denaturing thing that we were talking about earlier, that you can target the AT rich regions? That reveals the different areas where you can have replication forks set up. There are 3 additional DNA A binding sites that are called I sites, or initiation sites. And there's binding sites for proteins IHF and FIS. So all of these things are binding in this one small region. You don't necessarily need to know what the sequence is for the 9 base pair sequence, that's the R site, or anything like that. But you should know that we've got this DO region that's AT base pair rich, and that there are binding sites for DNA A, IHF, and FIS. Here's a list of all of the proteins that are required in initiation for E. coli at the origin. We're going to talk about these, but this table just summarizes their functions. So it's a good thing to have to just kind of glance at if you need to refresh yourself. So what we're going to see throughout is that there's a protein family, the AAA plus ATPase protein family. ATPase is associated with diverse cellular activities. And these proteins form oligomers, which means that there's a bunch of them all together that form an active enzyme, holoenzyme. And they hydrolyze ATP pretty slowly. So if their sole function was to be an ATPase, they'd be pretty bad at it. But what we see oftentimes is that the hydrolysis rate is actually a control mechanism. Think about it like a timer. And what the hydrolysis of ATP does is it mediates the interconversion of the 2 different states of the protein to pretty much say on or off. So the DNA A protein is a critical component, okay? It is a member of this AAA plus ATPase family, and when it's bound to ATP, it's active. When it has ADP in that active site, it is inactive. So you can again think about it as a timer. Okay, we've got ATP. We can bind some DNA and do some business. When it's inactive, it's not going to initiate replication. So in order for DNA A to be functional, you actually need 8 ATP-bound DNA A molecules, and they bind to the R and the I sites in the origin. So ORI-C, you see how it is italicized? We're talking about a gene or location on a chromosome. So once we have all of this binding, we're going to see some strand separation in that DO region. And the reason why this happens is because we've got strain due to positive supercoiling, and we've got all of these molecules binding the DO region. So between those two things, the strands start to separate at the origin. The next step is to load DNA B helicase. And to do that, we actually need an additional protein, DNA C. It's also a member of that AAA plus ATPase family, and it helps to load DNA B, which is the helicase, onto the separated DNA strands. DNA B is a helicase. It migrates along the single-stranded DNA in the 5' to 3' direction, which is the direction of replication. And the direction of the replication fork. And it unwinds the DNA. So it primes the way for the DNA polymerase. We've got single-stranded DNA binding proteins, or SSB, and that stabilizes the separated strands. Remember, to separate those strands, we are taking away all of the base stacking interactions and the base pairing interactions, which is highly unfavorable. And so to do this and stabilize it, we have to have proteins kind of filling in the gaps so that we don't have hydrogen bonds that are not made anymore, because otherwise replication wouldn't occur. If we couldn't stabilize this very unstable formation of DNA, we wouldn't be able to replicate it. We also have DNA gyrase, which is one of those topoisomerases that relieves the stress ahead of the replication fork. So if you've got, remember I'm no artiste, and then you've got this bubble there's going to be stress here if this is your replication fork. And you've got all your machinery here doing the business, replicating DNA. There's going to be stress here because you've got this open pocket where all of this stuff is bound, and you're separating the DNA strands much more than they would normally be separated in order to accomplish this. So that induces some stress up ahead of the replication fork that has to be relieved. So here's the model for initiation of replication. We've got DNA A binding, and that introduces some strain to the DO region. Once that happens, we have more DNA A binding, and we induce that denaturation of the DO region. This HTH, that's one of the domains of DNA A, and that stands for helix turn helix. And that describes a domain within DNA A that has a helix and then a turn and then a helix for its structure. And that's involved in, helping the different subunits to interact with each other. So we denature that DO region, and then we allow for IHF to bind. Then we've got DNA C coming in to join, and it helps to load DNA B on each of the individual strands. So where we're left is we've got this helicase that can help unwind And we've got all of these DNA A molecules and the addition of all these other molecules that are able to kind of help stabilize this open position. The helix turn helix region of the DNA A, I want to mention too, can also interact with DNA. So in places where we're still trying to unravel DNA, it will interact with the DNA. It can also be a part of the region that helps to allow for all of those different DNA A molecules to interact and be right next to each other on the same strand. So it's kind of double duty there. Initiation actually occurs once in each cell cycle. So we're not just constantly making DNA all the time willy-nilly. There's timing involved. And once you get that started, you're not going to then say, okay, halfway through, okay, we're going to initiate again and make some more. No. So HDA protein is yet another AAA plus ATPase that binds to the beta subunits of DNA Pol 3 and stimulates hydrolysis of ATP to the bound that's in the bound DNA A. And remember that DNA A has to have ATP in order for it to be active. So once it has ADP, it's going to disassemble. The binding of ATP to DNA A takes about 20 to 40 minutes for ADP to be released and ATP to bind. So not only is DNA A really slow with hydrolyzing that bond, the gamma phosphate, but it also very, very slowly releases its product. And all of that helps for maintaining the timing of replication. In the lab, if you've ever worked with E. coli, they double every 20 to 40 minutes. And this is partially due to the fact that DNA A is not available. It's still ADP bound, and it cannot bind the plasmid DNA in order to replicate. There's also some regulation of replication initiation via methylation. So there's methylases that will methylate DNA, adding a CH3. And they usually recognize a particular sequence, and they'll methylate a specific sequence or a specific nucleotide within that sequence. And so the DNA adenine methylation methylase will methylate the N6 position of adenine within this specific sequence, the palindromic sequence of the ORI-C. When that happens, we actually have hemimethylated ORI-C sequences. So remember that we're replicating the whole thing, including the origin sequence. And the old DNA which is the template, is methylated, whereas the new one is not. The hemimethylated ORI-C sequences, so meaning that we've got old and new, there's an interaction that occurs with the plasma membrane. And there's also the binding of a protein called seq-A. After seq-A dissociates, the ORI-C sequences are released, and then they fully methylate. So they methylate the other strand, and that ORI-C sequence is fully methylated, which allows DNA A to bind again. So that's another process that, one, takes time, And 2 is coordinated so that you're not initiating replication again before you want to. Then we've got elongation. So we've got everything set up, almost everything set up, and we need to elongate. Elongation means that we're going to start generating that leading strand and the lagging strand. The leading strand is pretty straightforward. You've got the primase that is going to make a short RNA primer at the replication origin. It has to interact with DNA B helicase. If it doesn't have that interaction, not going to get anything happening. The primase and the DNA B actually move in opposite directions. I know that's kind of interesting, but it still needs that interaction to know, so oftentimes you see this sequential addition of proteins and enzymes in these processes. And partially that's due to the fact that you need to have some kind of a way to recruit a protein or enzyme to the right location and orient it properly on the DNA strand in this case. And that's what's happening here. We're orienting the primase so that it can go and do what it's supposed to do. Once the primase drops down that primer, DNA pol 3 can add nucleotides to the primer terminus, and there we go. So the DNA pol 3 is actually linked to the DNA B that's tethered to the opposite DNA strand. Remember that there's helicase on both strands. And the DNA pol 3 holoenzyme, we've got kind of 3 separate arms that are all capable of doing polymerization and proofreading, etc. And we're attaching to both the leading and the lagging strands at the same time and chugging along. So you have to have a way of being tethered to move in sync along the DNA. The lagging strand is synthesized in short Okazaki fragments, and as the leading strand is being made, the primase will synthesize an RNA primer that DNA pol will add to, and then you have a short fragment made, then you have another RNA fragment or primer added on, and you keep going on and on, kind of alternating between the two. The lagging strand also loops around so that the one asymmetric DNA pol 3 dimer complex can synthesize both strands. So you've got one major huge DNA pol complex that can actually synthesize both strands at the same time. So DNA pol 3 uses one set to continuously make the leading strand. The other 2 core subunits are used to kind of cycle through and kind of leapfrog each other, making the Okazaki fragments. And there's a lot of words right now. Don't despair. There's a lot of pictures coming up too. So for all of you that like the words, here they are. Then we're going to look at the pictures and kind of put it all together and talk it through again. Here are the pictures. So this is how we synthesize those Okazaki fragments. We're focusing on this strand here. So the leading strand, that's the top one, easy peasy, not a problem. And we're only showing the core polymerase here. Remember, there's a ton of other things too. So the replication fork is moving this way. Leading strand is doing what it's doing. The lagging strand, remember, we're making our fragments in the opposite direction of the replication fork. So we've got the DNA B helicase that is helping to unwind this DNA and keep it apart. We've got the single-stranded binding proteins that are also stabilizing the single-stranded DNA. And we've got the primase that's making an RNA primer. What will happen next is one of the core polymerase subunits is going to bind here and kind of boot off these single-stranded binding proteins, and it will start to synthesize a new strand. It won't be able to go all the way to, this end here, and it certainly won't be able to seal the deal, close the nick. But what happens is once you make a fragment, an Okazaki fragment, you still got some of these single-stranded base binding proteins present to stabilize in between the fragments. And eventually, DNA pol 1 and the ligase will come through and stitch all of these pieces together. We've also got this clamp loading complex, and we haven't talked about that yet. The purple, this is the beta clamp. There's 2 beta subunits that come together and help the core polymerase to hold on to the DNA while it slides along. It increases the processivity of the enzyme so that you're not dropping off your DNA pol every X number of nucleotides, but you can actually stay on and synthesize thousands of base pairs. There's this clamp loading complex that is really handy for allowing for the binding and unbinding of these 2 subunits that kind of leapfrog each other in making the Okazaki fragments. And so we're going to talk more about that in a second. DNA B helicase is going to travel along the lagging strand and unwind the DNA in the 5' to 3' direction. And again, we're focusing on the lagging strand because there's a lot of coordination going on, and there's a lot of choreography that has to happen for all of these things to work in tandem. The DNA G primase will occasionally associate with the DNA B helicase and synthesize a short RNA primer. So there needs to be room for it to do that. You can't have a polymerase sitting there. You got to have some space for that primase to bind and do what it does. So let's look at the figure next to this. We've got the clamp loading complex that has an open clamp. So the clamp is 2 beta subunits, and it's primed and ready to add another one. Okay. We make a primer. We add on to that primer. We make an Okazaki fragment. Now we need to add on a new primer. So what happens is the primase will bind. And then we have to load on a new clamp and allow the core polymerase to bind again. So we're going to make a primer here. We make the primer, the polymerase will bind, and then happy day. The new clamp gets loaded on after the primase leaves. So that clamp that's open on the clamp loader will come in and attach to the template that was made by the primase. Once that happens, we can start synthesizing a new Okazaki fragment on the lagging strand, because now we've got something for the core polymerase to be attached to and slide along to make that next segment. Replication halts after you make an Okazaki fragment, and DNA pol 3 dissociates from the beta sliding clamp. So after a segment is made, the clamp is left behind. And so the old clamp is left, and you're able to synthesize a new fragment with the complex, the core complex that left it behind. So that's why we have kind of those two. There's only two core subunits shown, but there's really three here in all reality. But this is just so that the image isn't as cluttered. So you can take that core polymerase that was attached with this one, and now it is attached to this new beta clamp, and it's going to synthesize a new fragment. Once that happens, it's attached to that new clamp. We make a new Okazaki fragment, and the cycle continues. We also have to ready another clamp at the same time. So these are all the proteins that act at the replication fork, and this is during elongation. So you should know all of these proteins and their functions. You don't have to memorize how many subunits they have or their molecular weights, but you should know their names and their functions. So let's look a little bit more closely at the clamp loader. So the clamp loading complex is a AAA plus ATPase that binds ATP and the new beta sliding clamp. It's made up of several different subunits. You don't need to know what they are. ATP binding opens the ring at one subunit interface. So you've got this clamp that's like this. The clamp loader opens up one of the interfaces. So this is an interface. This is an interface. We maintain one interface, but we open the other. And that's what ATP binding does. Hydrolysis of ATP causes the clamp to close down around the DNA. So clamp opening involves ATP binding. To go the other way involves ATP hydrolysis. Again, think about this as a timer. And it can also be kind of like linked to the conformation of the enzyme. So you can have acceleration of hydrolysis when it's time, when there's DNA and the clamp is trying to interact with the DNA, then ATP hydrolysis can speed up. So these ATPases are really, what that function is a timer that allows for a particular function to happen at a given time versus it just happening and hoping that it lines up with all the other steps of replication. And we say that ATP hydrolysis is slow, but slow is relative. All of these things have timing relative to each other so that you have all of these things happening, in sequence and in the proper sequence versus randomly and hoping that they all line up. The final steps in lagging strand synthesis involve DNA pol 1 or RNase H1. Both of these enzymes are able to remove an RNA primer. The DNA pol 1 can also replace that primer with DNA. Once that happens, you still have a nick. And that has to be sewn together with a DNA ligase that uses either ATP or NAD to seal the deal. DNA ligase catalyzes the formation of a phosphodiester bond between the 3' end of one DNA strand and a 5' phosphate. So that is different from just making a phosphodiester bond. We're looking at making a link between the 3' OH and the 5' phosphate at the end of another strand. The phosphate actually has to be activated by adenylation, which uses ATP in viruses and eukaryotes, or NAD, which happens in bacteria. Again, you can see how spendy this whole DNA replication thing is. Lots of ATP being used. This shows the DNA ligase mechanism. So we've got first the adenylation of the DNA ligase. So we're going to adenylate a residue in the enzyme, and then once that happens, you can use that to then activate the nick. So we've got this nick that we need to repair. You activate it, and then you can actually have an attack that is very similar to the reaction that DNA polymerases use. And that seals the deal. Now that we've done all the elongation, we need to terminate. Replication forks meet at a region with multiple copies of a 20 base pair sequence called the TER sequence. And TER sequences are found near each other but in opposite directions. So this creates a site that the replication forks can't leave. So once the replication fork reaches there, that is the land of no return. TER is also a binding site for the protein TUS, the terminus utilization sequence, and that causes a replication fork to stop as well. There's only one TUS-TER complex that's functional per replication cycle. So you catch whichever replication fork reaches the finish line first, and then the other replication fork, when it reaches the replication fork that's already stopped, will stop too. Once we have this, and we've got our 2 copies of bacterial DNA, we actually have 2 linked chromosomes. They're wound around each other. And in order to separate them, we have to use a topoisomerase. And that's going to unwind the intertwined chromosomes and temporarily break and reform so that we can actually separate these plasmids. So replication in eukaryotes is similar, but it's more complex. And you'll see that as a trend as we move through RNA metabolism and protein metabolism. In yeast, replicators are the replication origins. They're also called autonomously replicating sequences, or ARSes. And yeast have about 400 replicators, and they span approximately 150 base pairs each, and contain lots of different conserved sequences that will recruit all of the things necessary to begin replication. Human chromosomes, which are very large, have about 30 to 50,000 replication origins. And this makes sense because, I mean, we've got a lot of DNA to replicate. And if we just had, a few sites where we can start the replication process, it would take a very, very long time to go through and replicate the entire genome. Ain't nobody got time for that. So there's additional regulation that occurs in eukaryotes, and this is linked to cyclins and cyclin-dependent kinases. This is our way of ensuring that replication of DNA only happens once per cell cycle. So just like the bacteria, we care about the timing and the frequency of DNA replication. It's a very energy intensive process, and we don't want to have multiple copies of our DNA running around in the same cell. That would be bad news. Or at least we don't want any more than is necessary. Regulation involves cyclins and cyclin-dependent kinases. Cyclins are actually small proteins, and they get destroyed at the end of M phase, which is mitosis. So these proteins are very tightly regulated, and as soon as they are finished being useful, they are degraded. You can imagine that this mechanism would be something that a cancer cell would certainly take advantage of and maybe upregulate how often you replicate. So there's timing involved, just like with the bacteria, how it takes time for things to regenerate. And, there are other processes that happen that prevent replication more than, it's not going to be more frequent than every 20 minutes or so. It's the same thing in eukaryotes. There are what's called pre-replicative complexes, which is kind of like the whole DNA A thing in bacteria. And that's established on replication initiation sites that don't have cyclins. So it's like the pre-ready. It's like, all right, all we need is the one thing and then we're ready to go. And their formation, which is sometimes called licensing, makes the cell ready or competent for replication. We've got 30 to 50,000 different sites that we want to initiate. So preloading these complexes all over the genome helps make, replication faster because we're already ready. We're just waiting for the signal. We're waiting for the light to turn green. The origin recognition complex is a 6-protein complex that recognizes and finds all these different origins throughout the genome, and it has a function like DNA. You'll see that there's a lot of similarities here, and of course one of the questions is going to be, what are the similarities, what are the differences? So just to kind of take note of all of these things. It'd be helpful to make a chart so that okay, this is homologous to this is homologous to that, and here's where they're the same, here's where they're different in terms of the processes. But in general, the rule is eukaryotic processes are typically more complex and involve more proteins, bigger protein complexes. Also, more regulation. Initiation also involves loading helicases, mini chromosome maintenance proteins, so the MCMs, and there's several of them. These helicases translocate 3' to 5' direction along the leading strand template, and it's homologous to or similar to the DNA B helicase. So let's look at a pre-replicative complex and see what it looks like. We've got the initiation site, which is bound by CDC6. And then we've got ORC as well, the ORC. And those things will also recruit CDT1. All of those things form a complex that then recruits the helicase. Once we have this pre-replicative complex, we have to activate it, and that's the green light. When CDC45 and the GINS proteins are available, they will come along and trigger the DNA that this complex is bound to denature, and it will also signal for that kind of pre-complex to fall off of the DNA and leave just the helicase. Then the remaining replosome components are added. You need DNA polymerase and some other things. You make that bidirectional replication fork, and then you get going. Another thing that you should think about is if you were trying to put all of these things together in a test tube to replicate the process of DNA replication, what are the components that you need? So that will help you think through the purpose of each of the different proteins that we've covered. And then if you could kind of recapitulate that in a test tube, what would it look like? So the rate of replication is approximately 50 nucleotides per second, which is abysmal in comparison to E. coli. And we compensate for this ridiculously slow rate by having a whole lot of origins. So that's why we need the 30 to 50,000 origins, because otherwise it would take forever, like hundreds of hours, to actually complete replication. And as you know, it doesn't take that long for us to do that. Let's look at the DNA pole. Okay, we've got several DNA poles in eukaryotes, and instead of being numbered, they have different, Greek letters. It's okay to not recognize them as long as you understand, as long as you can look at it and say, okay, I know what this one is, I know what that one is, I know what that one is. You'll be fine. So we've got a DNA pole that synthesizes the leading strand, very highly processive. It also has proofreading exonuclease activity in the 3 to 5 prime direction. We've got another pole that synthesizes the lagging strand. It also has the same kind of exonuclease activity that can proofread. Then we've got a DNA pole that also has primase activity, and it can synthesize the RNA primer and extend it out a little bit to initiate the synthesis of the Okazaki fragments. It does not have proofreading ability. So know these 3 different types and be able to compare and contrast them, know what their jobs are. We have a protein that acts as a circular clamp, just like the beta subunit in DNA pole 3 in E. coli. And ours is the proliferating cell nuclear antigen, PCNA. It enhances the processivity of the 2 polymerases that actually add on to the leading and lagging strands. We've got an analogous protein for the single-stranded DNA binding protein. Ours is the RPA, replication protein A. And its function is equivalent. It helps to stabilize single-stranded DNA. We've got a clamp loader, the RFC, replication factor C. And it will assemble active replication complexes. And it actually has significant sequence similarity to the subunits in the bacterial clamp loading gamma complex. You don't necessarily have to see sequence similarity, but you will usually see structural similarity, and that is oftentimes enough to have a similar function. Termination happens when the replication forks that are nearby converge. So they kind of crash into each other like bumper cars. There's a viral DNA polymerase that is actually a target for antiviral therapy. So when you get sick with a virus, that virus has the code to make a lot of proteins that it needs to replicate itself. And also it causes a response eventually in your body that makes you feel like, hot garbage. And one of the things that you can do when you're trying to develop antivirals or antibacterials is target proteins that are essential for the propagation of the infectious agent that will not affect the organism, or at least will have very minimal effect on the organism. And to do that, you can exploit differences in structures and functions that will inhibit one protein or one enzyme from doing its job while the organism can still thrive. And that strategy is used here. There are differences in viral DNA polymerases that can be exploited to make antivirals. Acyclovir inhibits DNA polymerase of the herpes simplex virus. And what it is a GTP, or sorry, it is a G base, guanine base. And instead of having this pentose ring, it is acyclic. So it's got all the components, but it doesn't form a ring. This can get converted to acyclo-GTP, so it still has the 3 phosphates, and it will competitively inhibit the DNA polymerases, but it doesn't really affect eukaryotic ones. So it will really only affect the viral DNA poles, and it will terminate replication because you can't do anything with this. You can't continue to add on bases after this. Now we're going to move into DNA repair. There are some sections that we are not covering in this, so if you like to look at your book, there's like a good portion that we are not covering from this section. Okay, so DNA molecules are clearly irreplaceable. We can't just make a new one to replace the one that got damaged. We have to actually repair them instead. So proteins, RNA molecules, those can be replaced because there's a template that you can use to just keep making them over and over again. But the DNA is the template. You can't replace the template. Mutations are linked to cancer, and this is something that you've already been introduced to, I'm sure, in biology classes. A mutation is a permanent change in the nucleotide sequence. There's different types of mutations, and again, you've probably heard of these too. You can have a substitution, where you replace one base pair with another. You can have an insertion, where you add in one or more base pairs, or a deletion, where you take some out. A silent mutation is a mutation that affects non-essential DNA, so it's something that doesn't have a gene product, or that gene product doesn't really do anything, or has negligible effect on gene function. Mutations very rarely give some kind of biological advantage. And most of the non-silent mutations are either neutral or deleterious. Non-silent mutations are ones that do have an effect on gene function and the gene product. There's a test that can look at the potential of a compound to promote mutation. And the thought here is that if a compound can promote a lot of mutation, then that means that it might be a mutagen, because it can cause your DNA to make, to have a lot of issues, right? And if it can do that to your DNA, then it could also potentially be carcinogenic. The Ames test, which was developed in the 70s, measures the potential of a given compound to promote easily detected mutations in a strain of Salmonella that cannot synthesize histidine. So you put down a little piece of filter paper in the center of a plate, and you try to grow your bacteria on this plate. So you have no compound, and that's your your negative control. So you don't have anything added. This is just how much of the bacteria is going to be able to grow just of its own accord. Mutation happening, homologous recombination, that kind of thing, allows for these bacteria to grow even though they typically can't. Then you add in your compound of interest, and you have different concentrations. So B is the higher concentration and B is the lowest concentration. I'm going to say that again because I think I may have said it wrong. So B is the highest concentration of compound, D is the lowest concentration. So we have a gradient. And what you do is you look at the ability of the bacteria to be able to make, mutations and grow on this media that does not contain histidine. If you can have all these mutations that allow for this gene to be intact again, and you can grow, and you can make your own histidine, then you'll see an increase in the number of colonies on the plate. And if you see that increase in colonies, then it suggests that the compound is a mutagen and potentially carcinogenic. So there's a lot of genes in the human genome that are dedicated to DNA repair. And all cells have multiple DNA repair systems just because there are so many reasons to keep your DNA pristine. If you have a bad template, then you're going to make bad everything else. DNA repair processes are extraordinarily inefficient when it comes to energy use, but who cares? If you're dead, then it doesn't matter, right? So you have to keep your DNA intact with as few mutations as possible in order to guarantee survival. So if the integrity of your genetic information is very low, then you're not going to survive very long. So using all the energy to maintain your DNA doesn't even matter. So this kind of goes against, what we were talking about before with all of these metabolic processes and how we're trying to conserve energy and, make sure that we're only using energy where we need to. We're going to regulate all these points. With DNA replication and repair, use it all, spend it all, black card, no limit. Because this is a very important process. Without it, everything else would cease to exist. That was kind of dramatic, I know. So let's talk about some repair mechanisms. We've got mismatch repair. And that is when you have a mismatch in your DNA where there's two bases that shouldn't be paired together. They're corrected to reflect the template strand information. So remember how we talked about methylation and how there's hemimethylated DNA when we've newly formed a new DNA. So the methyl groups on the template DNA help to identify it and say, hey, I'm the template, so if there's anything wrong, don't change me, it's the other one. So that will help with making sure that you're not introducing a mutation. So this is a summary of, the methylation and mismatch repair. When you start off with your template, you've got two old strands. They're both methylated. We do replication. And now we've got that semi-conservative process of replication where we've got one old and one new strand in each of the two new DNA molecules that we form. For a little while, you've got hemimethylation. which means you've got one strand that's methylated, one strand that isn't. The strand that isn't is the newly synthesized strand. So, if you have some kind of a mismatch, you're going to go to the strand that is not methylated and change that one. Eventually, that methylase is going to come in and methylate the other side. So remember that methylases typically recognize a sequence, and then they methylate a nucleotide within that sequence. And then you're not going to be able to tell these two strands apart. So mismatch repair has to happen here to make sure that all of the mismatches that are made during the replication process are caught. That doesn't mean that mismatch repair doesn't happen at any other time in the cell, but this is the time where you can tell the difference between the template strand and the newly formed strand, and you can rectify any issues by changing the newly formed strand versus changing the template. So here's the model for the early steps of methyl-directed mismatch repair. We've got mut S, which recognizes the mismatch. A mismatch usually introduces some kind of a bulge or different, it pokes out a little bit so that it doesn't look like the rest of the DNA molecule. And that mut S will recognize the mismatch. Mut H recognizes the same sequence that the methylase does, and it will bind where the methyl group is. We've also got mut L, which can bind to mut S at the place of the injury, so the damaged site. We've got a mismatch, and it will slide along and move to that mut H that's bound where the methylation is, and it will leave a mark by cleaving the unmethylated strand. So mut H will cleave the non-template strand to mark it to say, hey, this is the one that needs to be changed. The unmethylated strand is then unwound and degraded from the point of that nick all the way through the mismatch site. And then you replace that DNA with new DNA. You need single-stranded binding proteins because that's going to stabilize the single strand, the template strand that's just kind of hanging out there. And then you've got an exonuclease, which is going to chew up DNA. You've got DNA pol 3 and DNA ligase. So a similar complement of things used in replication is also used in mismatch repair. Mismatch repair on the 3' side of the cleavage site is very similar to issues on the 5' side. So remember that it matters which side the cleavage site is on. And you're going to have different exonucleases because exonucleases are directional. And they only will, in general, they will only chew up DNA in one direction or another. Or nucleic acids, I should say. So once we have our nick that's caused by mut H, if we have something that is on the 5' side versus the 3' side, that will determine what happens. So if we've got the nick over here, this is the 5' side. We're going to chew up everything from here all the way up to the place where there's the mismatch. And then we're going to use pol 3 and DNA ligase to seal the deal and remake that DNA. If it's on the opposite side, the 3' side, then we're actually going to chew up everything over here and go 3' to 5' and chew it up. Once we have that happening, then we can go in with DNA pol 3 and the ligase and everything and fix it. Mismatch repair is costly. You are degrading large portions of your DNA and then redoing the work. So that is a lot of energy investment to make the DNA and then energy investment to fix it. There are lots of proteins that are structurally and functionally analogous to the bacterial proteins involved in mismatch repair for eukaryotes. A lot of the details are actually unknown. We don't know exactly how the newly synthesized DNA strands are identified, but it does not involve the same GATC sequences that we see in bacteria. So this is kind of a reminder that everything is not understood. And so textbooks sometimes represent science as this, oh, we know everything and this is what it is, and read it and memorize it. It's not like that at all. There are still lots of openings for fresh research to progress, and then we fill in the gaps as we can. Another type of DNA repair is base excision repair. And this requires DNA glycosylases. And they recognize lesions and remove the affected base. And they just leave like the part of the phosphodiester bond. So they remove the base, but the sugar and the phosphate are still there. There's lots of different DNA glycosylases, and there's usually a specific glycosylase that's associated with a specific type of lesion. When a glycosylase takes off that base, it leaves what's called an AP site or an abasic site. AP site stands for apurinic or apyrimidinic site, and that just means that you took the base off. There are uracil DNA glycosylases that specifically remove DNA that has uracil that results from the spontaneous deamination of cytosine. So remember, our DNA undergoes lots of damage. And sometimes what that means is you have spontaneous deamination and other things that occur to cause dimers to form, all types of craziness. And this glycosylase does not remove uracil from RNA. And it doesn't remove thymine from DNA. It simply removes uracil from DNA. So it is very specific for what it does. There are other DNA glycosylases. And I should mention that there's a lot of uracil glycosylases because that is, the deamination is something that happens quite regularly. And you need to be able to recognize and remove that. And there's some thought that there's, thymine in DNA versus using uracil and RNA, simply because if you have spontaneous deamination occurring, then you could change the template that you're passing down from cell to cell. So by using a different base, you now can detect spontaneous deamination versus just a base. And we'll talk more about that in class. There are other DNA glycosylases that recognize other forms of damage, other lesions. Purine oxidation, adenine deamination, alkylated bases, pyrimidine dimers, all kinds of things. In bacteria, when we have the deoxyribose 5-phosphate left behind at that AP site, it's removed and it's replaced with a new nucleotide. So there are AP endonucleases that cut the DNA strand that have the AP site. And then DNA pol 1 shows up again and replaces the DNA. So you can see why DNA pol 1 in bacteria, why it's so abundant, because it has a lot of work to do to repair DNA. Then we've got DNA ligase, which seals the deal. It seals the nick that remains after DNA pol 1 comes and replaces the nucleotides. So this is a little figure that shows base excision repair. We've got a DNA glycosylase that's going to remove a base. So you'll see here, one of these things is not like the other. One of these things just isn't the same. It's a GU base pair in DNA. This should not happen. Why did it happen? Because the cytosine that was there got spontaneously deaminated, and now it's a uracil. So a uracil glycosylase comes along and says, oh, that's wrong. I'm going to cut out this uracil. So now we've got this AP site. A basic. The AP endonuclease recognizes this site, and it's going to cut right here. Now we've got this nicked DNA, and we need to do something about it. DNA pol 1 will come along, and remember that it has that 5' to 3' exonuclease activity in one of its subunits. That's kind of the front of the molecule. It's going to come along and chew up a piece of this DNA here. So it's going to chew it up, and then as it's chewing it up and moving along this piece, it's also going to synthesize a new piece to fill in and fix the A basic site. So now we have our fixed base pair, and we've got this new strand here, but we've still got a nick because the DNA poles cannot take a 3' hydroxyl and catalyze the reaction to take the 5' phosphate of the next sugar base combo and link them together. So that's where the DNA ligase comes in, and it seals the deal, and now we've got a repaired DNA. So you could see why DNA pol 1 may not necessarily need to be as processive as DNA pol 3, because we're not making an entire genome. We're just trying to repair some things by and large, or trying to replace shorter segments of DNA, or we're getting rid of an RNA and then replacing that short segment with DNA. So you can see why it doesn't need to hang on to the DNA as well as DNA pol 3 does. The next type of repair that we'll talk about is nucleotide excision repair. And that is repairing a lesion that is causing a big distortion in the overall DNA structure. What's required for this is a multi-subunit enzyme called an nuclease, and it hydrolyzes two phosphodiester bonds, one on either side of the distortion. So it makes two cuts. DNA pol 1 in E. coli or DNA polymerase epsilon in humans will fill in that gap. And then you have DNA ligase sealing the deal. You can see the trend here for repair mechanisms. You have identification. There's some method of identifying that there's a problem. And you mark it by cutting, or you can use something to guide you, like methylation, to say, oh, okay, we're supposed to fix this side. But there's some kind of identification and labeling of the place that's going to be repaired. Then you have a cut and removal of a base or a section of the DNA strand. You replace it with a DNA polymerase, and then you seal the deal with the ligase. So the overall steps of all of these repair mechanisms are pretty similar, but what they fix is different, and how they recognize these different problems is different. So here's the comparison of E. coli and humans. We've got this DNA lesion that's causing some kind of structural change in the DNA. And we have the exonuclease coming in and binding. You can see that there's a nick here and here that's introduced. The nicks on either side of the lesion, we make a smaller, we release a smaller piece of DNA in E. coli. So this piece that gets liberated is about 13 nucleotides, whereas in eukaryotes and humans, it's like 29 nucleotides. And a helicase is what helps to kind of unwind that DNA and allow for the removal of this oligomer. Then you've got the polymerase coming in and replacing the DNA that we just exited, and the ligase comes in and seals the deal. That's it for Chapter 25. Like I said, we're not covering homologous recombination, but if it's something that's interesting to you, you can go ahead and read it for yourself. But we have a lot of things to cover in this class, and we just don't have time for everything. Again, focus on the similarities and differences between eukaryotic and bacterial replication. Look at the analogous proteins, understand the functions, and be able to kind of boil it down to what is the process. Overall, what's happening here, so you can draw similarities to RNA metabolism and protein metabolism as we move forward. Thanks for watching, and stay safe.